In the previous session, we discussed about protein structure and denaturation process. In this session, we are going to discuss about isoelectric pH of proteins. Definition: isoelectric pH, which is designated as Pi of a protein, is defined as the pH at which the protein exists as zwitter ions. Zwitter ions are dipolar ions, meaning carrying equal number of positive and negative charges. So, at isoelectric pH, the net charge of proteins is zero, so they will not move under an electric field, and since the solubility is less, they tend to aggregate and coagulate and get precipitated. So every protein is having its own isoelectric pH. The proteins which are having amino acids, amino containing positive charge, and acid carboxylic group contains negative charge, and the protein contains varying number of positive or negative charge. They may not be equal in number. The number of positive and negative charge, when they are subjected to certain pH, they can get the equal positive and negative charge. That pH which makes protein to get equal positive and negative charge is called as isoelectric pH of that protein. And in that pH, the proteins are less soluble and hence get aggregated and precipitated. This is the meaning of isoelectric pH. The properties: proteins have least solubility at isoelectric pH. At isoelectric pH, proteins carry no net charge, and thus they exert minimum electrostatic repulsion. So there is less repulsion, means they are less soluble. So at isoelectric pH, proteins tend to aggregate and precipitate. The few examples of isoelectric pH of certain proteins are listed here. Isoelectric pH of pepsin is 1.1, casein is 4.6, albumin is 4.7, and human hemoglobin is 6.7, and etc. Next is related to this topic. There is one more heading called as isoelectric precipitation of protein. Precipitation of protein at their isoelectric pH is called isoelectric precipitation. Some proteins are immediately precipitated when adjusted to their isoelectric pH, and the best example is casein, which gets precipitated immediately when pH of casein solution is adjusted to 4.6. Isoelectric pH of casein is 4.6. You can see in the above example also it is quoted casein pH of isoelectric pH is 4.6. If you take the milk which contains this protein casein, and if you decrease the pH of the milk, which is usually above 4.6, by adding lemon juice or acetic acid or any other solutions, and whenever the pH reaches the 4.6, the milk will get curdled, and proteins will get separated, and water will get separated, and making the casein protein in the milk to precipitate. This is a method is used for certain dishes preparation, especially sweets like making rasgulla or paneer. The milk has to be curdled. For that, they will add acetic acid or vinegar and make it curdle or casein to precipitate, and from that they will prepare these types of milk products. And next we are going to discuss about test for amino acid and proteins. The test for first amino acid, that is nitrogen reaction. It is a general test for detection of compounds having free alpha amino acid groups, that is free amino acids and peptides and proteins. They will react with nitrogen. This is one of the most sensitive tests for amino acids. Procedure: take a sample and add freshly prepared nitrogen solution, and if the sample contains amino acids or protein or peptide, observation: you can see a violet color is obtained. The sample giving violet color indicates the presence of protein. If the violet color is not obtained, that means the given sample is not a protein. If you tell that milk contains casein and egg contains albumin, and you can believe only after doing this test, you can take the samples and add nitrogen reagent solution and boil for few minutes, and obviously you will get a violet color. And if you just take a water, tap water, which is not a protein, you can add nitrogen and then boil it. You will not get violet color because water doesn't contain protein. If you take egg, albumin, or some of the other protein containing example items, then you will get violet color indicating the presence of amino acids. Principle is given below. Ninitrin reacts with free alpha amino acids amino group of proteins to give a blue or purple colored complex called Riemann purple. So the reaction process is given in the illustration. How amino acid reacts with nitrogen and ending up with the last you can see remains purple. Next is we'll discuss about test for proteins. The heading is test for protein is biuret test. This test is given by all proteins, hence it is used as a general test for proteins. Procedure: take a few sample and add copper sulfate solution 1% usually, and add 10% sodium hydroxide NaOH solution, and a violet color is obtained means the sample contains protein, and if no violet color means the protein is absent. Similarly, in this test also you can take the white part of the egg, that is egg albumin, in a test tube and add the copper sulfate solution and then NaOH. Shake it for few minutes and you will get a violet color, indicates it contains proteins. And if you take water, tap water is not having protein. If you add this copper sulfate and NaOH, you will not get violet color. It will be blue color only, because copper sulfate is blue. You will get, it will end up with blue color only. If the sample contains proteins, it will end up with violet color. The principle of this test is given below. Cupric ions in an alkaline medium forms a violet color complex. Cupric ion in an alkaline, because sodium hydroxide is added, forms a violet color complex with peptide bond because it is present in proteins. The minimum requirement of positive biuret test is the presence of two peptide bonds in the molecule. Peptide bonds means it is related to protein that reacts with cupric ions in alkaline medium, thus giving the violet color, indicating presence of protein. Next is plasma proteins. Let us discuss what are plasma proteins and what are their importance or function. So before going to the plasma protein, let us see what is plasma. So blood is made up of cellular, acellular components, and these cellular and acellular components which makes the blood also contains certain factors such as clotting factors. So the whole blood if you take and then in presence of anticoagulants it is centrifuged, then you will get different layers. The bottom precipitated red color, you can see the cellular part, and then you will have a platelets, and then in the top you will have plasma which contains proteins and other molecules along with clotting factors. Plasma is the liquid cell free part of blood that has been treated with anticoagulants. What is serum? Serum, if you take the blood and then in absence of anticoagulants, if you leave it for certain time, it will clot, and the clotted blood settles in the bottom, and then it is having a liquid part that is in the above is free from anticoagulants, that is called as serum. Serum is the liquid part of blood after coagulation, therefore devoid of clotting factors, as for example fibrinogen. So non-clotted blood centrifuged, the topmost liquid part that was obtained which contains clotting factors is called as plasma. If you take the blood and clot it, and then the liquid part obtained is called as serum. Serum contains fibrinogen. If you see in the bracket, serum is nothing but plasma minus fibrinogen. Next is albumin. Albumin is the major plasma protein. Coming to the plasma protein, plasma contains hundreds of different proteins known as plasma proteins. Normal total protein concentration in the plasma is 6 to 8 grams per deciliter. All plasma protein except immunoglobulins are synthesized in the liver. So most of the plasma proteins are synthesized in the liver. All plasma protein except albumin are glycoproteins. So there is a big list of plasma proteins. In that, the very important plasma proteins are listed in the below. You can see albumin, globulin, fibrinogen are three important classes of plasma proteins. Albumin constitutes for 3.5 to 5 gram per deciliter of blood, and then globulin 2.5 to 3.5 gram per deciliter, fibrinogen 200 to 400 milligram per deciliters. This is the quantity of plasma proteins. Significance: decreased plasma protein concentration is an indicator of liver disease, because you can see, yeah, we have discussed that most of the plasma proteins are synthesized in the liver. If the plasma protein level is less than the normal, it indicates liver disease due to defect in the synthesis of albumin and other plasma proteins, or nephrotic syndrome, that means kidney disorder. In that also the plasma protein is less due to abnormal excretion of albumin in the urine. So this indicates organ diseases, and hence plasma protein is an indicator of organ disease like liver and kidney. Albumin level is also decreased in nutritional deficiency like kwashiorkor, burns, and in diseases like tropical sprue. So it also indicates certain diseases. Edema is the consequence of hypoproteinemia. So protein is less, then it will lead to edema or hypoalbuminemia. Edema means accumulation of fluid in the tissues. Hence plasma protein, their normal quantity, its importance, and the abnormal quantity and its indication is going to help in detection of diseases and suitable treatment. So as we discussed about the three plasma protein important, let us discuss one by one. Albumin. Albumin is the major plasma protein. It constitutes up to 60% of plasma proteins. So in the plasma proteins, this is the abundant protein. A human albumin has 585 amino acids. It is synthesized in the liver. So albumin is having 585 amino acids and is synthesized in the liver. Globulins. Globulins constitute many different proteins which are separated into four distinct bands in electrophoresis. Electrophoresis we will discuss, that is the movement of charged molecules in an electric field. We will discuss in detail later. Each band consists various globulin proteins, and globulin fractions are given below: alpha 1 globulin, alpha 2 globulin, beta globulin, and gamma globulin, and the example is listed or quoted. This is about globulins. Next one is fibrinogen. The third plasma protein. Fibrinogen are proteins that are responsible for clotting, coagulation, blood clotting, and these three important plasma proteins is very important among all other proteins present in the plasma, and remaining we'll discuss in the next session. Thank you.